last student sensor rate seminar series. My name is Zeng. I'll be the chair of today's talk, and I'm also one of the members in the student-led research interest group. The primary aim of our sensor rate group is to share research in the area of sensor and sensing technologies. We hope the student seminar series will enable knowledge shared and foster collaborations. Please visit our website to explore more about our organization. If you'd like to present your research or interested in joining our group, please do not hesitate to email us. Okay. The title of today's talk is Differential Sensing of Mitogen Activated Protein Kinase Isoform. We're very excited to have Dr. Zeng here. Dr. Zeng is a postdoctoral fellow in the Department of Chemistry. She completed her bachelor's degree in chemical biology in 2013 and PhD degree in analytical chemistry in 2018 at Wuhan University. In 2016, she joined Dr. Eric Ansland's research group as a visiting scholar and then continued her postdoctoral research till now. Dr. Zeng's research interest mainly focused on developing differential sensing arrays and performing chemical data analysis using machine learning techniques. Before the talk begins, I'd like to remind you that you can ask questions at any time by using the chat box. We'll collect the questions and ask the presenter at the end of the talk. Now, I would like to hand over to Dr. Zeng, and the time is yours.
Okay, let me share my screen. Okay. Okay.
Dr. Zeng, you are muted. Yeah.
Okay.
Yeah.
Good. So, yeah, thanks for inviting me to this seminar. It's my honor to present our group's differential sensing research, which has been studied for nearly 15 years. Today, I'm going to talk about the sensing of the isoforms of mitogen activated protein kinases. So, why do we focus on the MAPKs? It is because the MAPK triggers a lot of important cell signaling pathways, thus regulating a series of cellular responses. And the abnormal changes in the activity is associated with many human diseases. And MAPKs are classified in 3 major groups, and each of them has several isoforms. Since the different isoforms have special contributions to the certain dysfunction, the testing methods of the individual isoform are very important. To study the kinase activity, the most common methods are the Western blot and HIC, but they do not measure the kinase activity directly, and their procedures are quite complex. By contrast, the optical-based sensing method could overcome the above limitations. For example, the Clatian-enhanced fluorescence strategy is a leading approach in this area. They can report the phosphorylation events with some selectivity, but it requires the specific receptor for each of the kinase target. Since we have more than 500 endogenous kinases, it's very hard to identify the biosensor's selectivity. Also, they report the fold change of the fluorescence intensity, so they do not measure the absolute quantification of the target kinase. But differential sensing could solve these problems. We are thinking to use the cross-active sensors instead of the highly reactive ones and highly selective ones, then using the chemometric tool to get the information, which is based on the pattern-based recognition approach. Here I have to say the algorithms or methods we use in chemometrics is just the machine learning, which is very popular nowadays. The tools our group use most are the principal component analysis, linear discriminant analysis, and the support vector machine. For example, we successfully differentiated the terpenes in the perfume by using the serine albumin receptors. Also, we developed some sensing enzymes to pattern the 20 glyceride samples. In our group, the most recent target in our differential sensing project is about MAPK. There are 9 isoforms from these three MAPK families, ERK, JNK, and P38. There are major two kinds of sensors we have developed. The first one is in situ generated chemosensors. The other one is SOX peptide-based biosensors. At the beginning, our group developed these in situ generated chemosensors and successfully differentiate these 4 isoforms in the cell lysates. Then we started making the peptides biosensors with the SOX chlorophyll, which is what I want to talk in detail today. So the docking site right here in the peptides could determine the selectivity for the different MAPKs. The cysteine next to the phosphorylation site of SOX chlorophyll is attached. Once the peptide was phosphorylated by the active kinase, a fluorescence structure will form in the assistance of the magnesium ion. We can detect the fluorescence signal and then collect the kinetic plot for the data analysis. At the beginning, we constructed a cross-reactive sensor array with 5 SOX peptides. Each of them has different affinity to the 3 groups. For example, the sub-D peptide, they can react to all the 3 families. And this one can react only with 2 of them, but this one only react with J and K. So the NFAT4 peptide, they have very low signal to the ERK1, but has significant signal to the JNK2. Then we put the peptide array with the 9 MAPK isoforms and get the fluorescence changes and the 20 minutes, along with the calculated rate constant. Using those data as the input for the chemometric analysis, here is the LDA score plot showing the in vitro differentiation of these 9 MAPK isoforms. And from the PCA biplot, we can see the contribution of each peptide to the certain kinase group. Then we perform the quantification of ERK1 in the presence of JNK and P38. The LDA plot shows that we can differentiate the ERK1 concentrations, and the SVM regression built the calibration curve, and we can get a good prediction to the samples with unknown ERK1 concentration, like here, these 2. Here are the results about the quantification of ERK inhibitor concentrations in the presence of all the 3 MAPK groups. Similarly, the prediction plot was built for the quantification of this inhibitor. In previous study, we used the recombinant enzyme, which cannot reflect the endogenous activity. In the following work, we expanded the application of this peptide array to quantify the ERK activity in both the cancer cell lysates and the tumor extracts. It should be noted that we achieved this goal without the need to suppress the related kinase activities. For the quantification purpose, here we only select the peptides that can respond to ERK, these 3. And the cancer cell was pretreated with the ERK inhibitor. Then the different concentrations of purified ERK were added to build the calibration curve. Using the delta fluorescence and the rate constant data, the LDA plot was generated here, and the SVM regression model was built. Since the ERK level is closely related to many other MAPKs, we try to activate or inhibit the actual ERK activity by different treatments. Under the impact on the ERK or JNK pathways, one peptide has various response to these treatments, like this. Using the same standard for collecting the input data, we can generate the response pattern of the different treatments. Then we use the previous regression model to calculate the ERK concentrations in these samples. then compare our prediction to the Western blot results, and we can find the results are quite similar. Finally, we use this peptide array to quantify the ERK concentration in the tumor samples. These samples were extracted from the mice bearing the tumor. The calibration curve was built by adding the purified ERK into the tumor extract and treated with the ERK inhibitor. Then we calculated the ERK concentrations in the tumor samples, which is also very close to the Western blot result. Based on all the work we have so far, there's still some room for the improvement, and we want to go further. This is what I'm doing since I joined the group. As you can see, this pattern by the original peptide array with 5 SUX peptides, the cluster of ERK1 and 2 are pretty close, these two triangles. Also, we have some issue to differentiate the 3 JNK isoforms, like the 3 groups of dots, and the P13 beta sample looks quite overlapped with them. So we want to make them separated better. So we rationally designed more peptides with additional selectivity to ERK and P38 groups. We expect to see a better differentiation performance within the ERK and JNK groups, also between the JNK and the P38 groups. And here we have some achievements. The 2 ERK isoforms right here, the 2 dots, they're still a little bit close to each other, but we can distinct them. It's hard because they have more than 85% identity and an amino acid level. And the 3 JNK isoforms are well separated in this space, the yellow, olive, and the green one. And more surprisingly, we got a very great differentiation of all the 4 P38 groups. Currently, I'm working on sensing the activity of the 3 groups simultaneously, which means within one sample, we are no longer only change the concentration of one of them, but 3 of them together. Okay, that's it. At the end, let me express my gratitude to my advisor, Eric V. Ansling, and all the past and the present group members. Also, I want to say thanks to Dr. Dana Zamora Alves, the former Ansling group member, and also thanks to our collaborator, Dr. Delby and Dr. Cowd, the experts in the MAPK studies from the College of Pharmacy. Well, any questions are welcomed.
Okay, thank you for your amazing presentation. Let's wait a few seconds for the question to come in.
Mm-hmm.
Okay, you can either unmute yourself and ask directly or type in the chat box.
Hey, Dr. Zeng, this is Mega from College of Pharmacy. Nice presentation, a lot to learn. So I have this very naive basic question for you. So the biosensors was you developed for MAP kinase. Can these be developed for other targets as well? Is it possible?
So you mean other kinase or some enzyme not kinase?
Yeah, like other kinase. So I work on FGFR receptors, but the main issue what we have with those receptors is the phosphorylation site is same for all the receptors. So do you think these kind of biosensors would help us to specifically identify those sites respective to the receptors?
I think so, because when we designed the peptides, we borrowed the reported, how do you say that, the substrate peptide for the MAPKs. So they reported the specific sequence for the docking site, and also for the phosphorylation site and the fluorophore, they have some certain like distance to design them. So I think there should be some reference for this.
Great, thank you.
Mm-hmm.
Right. I have a question about how stable the molecules for practical sensing applications.
You mean for like the replication, the repetition, or how stable for the biosensor itself?
Yes.
Oh, I think the peptide is quite stable compared to like antibody. Yeah, because we store the peptides in the freezer for more than 1 year, and the fluorescence looks not change too much.
Oh, okay.
Mhm.
To follow up on that last question on stability, so like after you put it into a cell, like how long does it still work as an active biosensor in the cell?
So actually, we just add the peptide into the cell lysates, not directly into the cell, but if the peptide can react very well with a certain activated kinase or the cell lysates pretreated with the activator, we can still observe an increasing signal. So yeah, I think it just still work very well.
If I could ask another question. So yeah, so I did see that you put in vitro, but I wasn't sure if you meant like cell culture in vitro or lysate or something like that. So is this something that could work in a cell or in a living organism, or is this something that is more tuned for just like for in vitro stuff?
So till now we're just using the intro stuffs, and we were thinking to use the patient sample and to test the tissue samples. But for the living samples, I think might be the chlorophyll. The chlorophyll can be an issue because it was excited by the blue light and emit at a green light. So I don't think it should be sufficient enough for bioimaging. Yeah.
Okay, and anyone have any questions? Okay,
Mr. Lee, I do have a question.
Okay, please go ahead.
So great presentation. I think I would have to study this quite a bit for me to answer this question, so I'm just going to ask it. Is this an advancement in doing the protein analytics or protein analysis of, say, specific or cancer tumors that could be utilized for, say, personalized medicine, where you're looking at a cancer tumor and then treating these cells or the cancer tumor with different medical treatments, medicines? Or is it not that far along yet? Or is this not the topic, not the appropriate topic for this conversation?
Actually, it is our topic because
okay, thank you.
Yeah, the reason for us to do the differential sensing and detect the different isoforms, for the precise medicine.
So I don't mean to take up all the time. Maybe we could talk offline. I'm interested in seeing and talking with you or the group or your professor about whether this is getting close for commercialization. Companies like Illumina or Cepheid might be interested in integrating this into their product line. These are publicly traded companies you're probably aware of. My point is, I know these executives at this company. I know a protein seek from Andrew Ellington from 5 years ago when I worked as an entrepreneurial advisor. for Dr. Bob Metcalf. And I'd like to kind of bridge the gap here and see if there's a way I can help get you either funding or closer to commercialization.
Okay, thanks.
We can follow up later.
Yeah.
Okay.
I think we can exchange our email after the meeting, after the talk. Okay, so due to the time constraint, we all have, if your question is not answered or you have further questions, please just email us and we'll deliver to Dr. Zeng and get back to you soon. I would like to thank you again for your great presentation, and I wish you all the best in your future work. I'd like to thank our audience as well for joining us today. This is our last seminar for the fall semester. Our next talk series may start on spring semester, which will contain many exciting information and research. So please remember to subscribe our list and obtain the latest notification of our upcoming events. We're very much looking forward to seeing you next time. Thank you.
Thank you. Bye.
Bye.